Hello, everyone. My name is Dr. Clement Chimesi. I'm a medical statistician at the Norfolk Department of Medicine at the University of Oxford. Today, I'll be giving a brief talk entitled Survival Analysis Techniques in Clinical Trials. We'll be moving from more traditional methods to more generalized versions known as multi-state Markov models. My talk will be very brief. So in view of that, I'll be giving top-level summaries of the underlying survival techniques. Thank you. So what is time to event or survival analysis? So these kind of statistical models are used in situations where we are modeling outcomes of outcome variables that are quantifying the time until a certain event occurs. And that event could be death, it could be infection, it could be even adverse reactions or adverse events in a study. And so it means that if you are even looking at time to, let's say, adherence or time to first infection, time to mortality, these are the type of outcomes that survival models seek to predict or seeks to model. Now, why did it get the name survival models? Pioneering works in this area were targeting studies that were looking at time after treatment till death occurs. And so it means that earlier works looked at modeling how individuals survives over time. And that is why it gave the name survival models or survival analysis. It doesn't mean that every outcome or the kind of outcomes you might use in your respective studies will be a survival outcome. So that is why a more generalized, better version is called time to event outcomes. So it means these are class of analysis used to model time to event outcomes. They are basically broadly three methods. So we have the non-parametric version, we have the semi-parametric and the parametric version. The non-parametric and the semi-parametric version, they are more relaxed in terms of certain parametric assumptions about the outcome. Specifically for the Kaplan-Meier, it doesn't make any assumptions about the covariate or any assumptions even about the outcome itself. And then for the Cox-Proportional hazard model, which is a form of regression, with the Kaplan-Meier, it looks at only one covariate at a time. So it means that situations where you want to compare survival times or survival curves between, let's say, gender, between males and females, or between different age groups, that is where Kaplan-Meier comes in. So it means for the Kaplan-Meier method, it only focuses on one covariate at a time. But for the Cox-Proportional hazard, it extends the Kaplan-Meier to include several other covariates in the model. Okay, so that's what we mean by the Cox-Proportional hazard model. But these two are known as unstratified models. But there's one particular assumption about the Cox-Proportional model. And the assumption is that, first of all, we assume that the baseline hazard rate should be constant or should remain the same for each individual. And another assumption, which is very important, that talks more about the constant hazard proportion assumption, is that the covariate, the effect of each covariate on the outcome must remain constant or the same over time. This is to say the hazard ratio between different groups, let's say different groups of a particular variable, should remain time invariant, or should not be time varying, or should remain constant over time. And it is not always the case that that assumption is met. And there are ways to statistically explore that. Now, for the parametric models, these are situations where you make some strong and specific parametric assumptions about your outcome. For instance, you impose a particular parametric probability distribution on the outcome, or you have enough evidence to suggest that your survival times or your time to event outcome will follow a certain probability distribution. And it also allows for some parametric assumptions on your covariate. And situations where the constant proportion assumption, that is to say that the covariate or some covariates do not have the effect of the covariate on the outcome do not remain constant over time, then the parametric methods could be ideal in that case. But there are other ways of handling them, which you'll find out very soon. So this brings us to what we mean by stratified and unstratified survival analysis. So unstratified survival analysis are considered when the constant hazard proportion or the proportional hazard assumption is met. If they are met, or if it is met, then the kind of analysis you do are known as the unstratified analysis. But situation if some of the covariates or some of your predictors, for instance, your categorical predictors, if some of them violate this assumption, then you can stratify the analysis with respect to those variables that violated that assumption, and we'll look at how to do that. So, in briefly, on stratified survival analysis are the standard Kaplan-Meier and the standard cost-proportional hazard models we know. But within the cost-proportional hazard model, any covariates that violate the constant hazard assumption, okay, we can stratify those covariates in that model. So it means that we can have a stratified and an unstratified cost proportional model. And for some common parametric survival models, although this presentation is not targeting much about the parametric models, and most clinical studies tend to use these non-parametric and semi-parametric models, there are certain parametric models you should be privy to or you should be aware of. So we have the exponential models, the Weibull models, log normal models, and so on. And these models are used when you make and you know some strong parametric assumptions about your time to event outcome. There are other considerations for which those parametric models too might be much more ideal or better than the parametric or semi-parametric versions, and we'll talk about that briefly. There are some relevant disclaimers that I think my audience should know. So you notice that for the non-parametric Kaplan-Meier method, it assumes that you are comparing the survival curves between a particular categorical covariate or predictor at a time. So it means you cannot look at or compare how survival times or survival curves changes between more than two covariates or more than two predictors. And that is where the Cox proportional regression model came into being to extend that. Fine. But because of that extension, because the Cox extends the Kaplan-Meier method, most published works assumes that the Cox proportional model is a multivariate survival model, which is very wrong. The right wording is that it's multivariable model. The reason why it is not multivariate is that in statistics or in regression analysis, we only say a regression model is a multivariate model, or a survival model could only be a multivariate model when you are jointly modeling more than two time to event outcomes, or when you are modeling two or more time to event outcomes. But the moment you are only modeling one particular time to event outcome and regressing it on several predictors, it doesn't make it a multivariate method. So that is something I want us to note. And there are also instances where people are making use of some form of survival outcomes, and which are not time to event outcomes. And because of that, they still assume that it's a survival analysis. In stricter sense, that's not the case. So if you are modeling, let's say, incidence of an infection, and where the data are count data, okay, you're looking at number of maybe individuals infected over a period of time or over time, and then you're making use of Poisson class of models, or you are modeling infection status, or let's say mortality status, and using logistic regression model by like let's say binomial logistic to estimate odds ratios, or log binomial regression to estimate, let's say, risk ratios, or use Poisson models to estimate incident rate ratios. Doing these or fitting these kind of models do not constitute a survival analysis. They're in stricter sense because they are not, the outcome is not time to event outcome. So that's one thing I want us to understand. You could be modeling a survival outcome, which is binary, but it doesn't mean you are doing survival analysis. So let's be very careful about that. So the choice of word is very important. So here I've given key differences between survival, the standard survival analysis, and this class of model. That's the Poisson or logistic regression of some kind of survival outcome, which is not time to event outcome. So you can get access to this particular. I'll give this slide to be made available, and you can always jot it down. So it means these are just the key part, or the key point is that the outcome must be time to event before you can classify it as a survival model or survival analysis. That's what I want us to note. And one thing too is that the logistic or Poisson models are not able to handle censoring. And censoring is something that we often even need to achieve before we can do survival analysis. This is to say, within your study period, not all individuals would experience the events of interest. So it means that if you're looking at, it means that if you're looking at time to death from a certain infection over a certain period of time, individuals who experience the event are uncensored, but those who remained alive within the study period become censored at the final time point. And basically, those individuals who did not experience the event within the study period are known as positive censored. And positive censoring is a key aspect or a key assumption that needs to be satisfied in order to use the traditional survival models. And that cannot be dealt with in situations where the data is censored. You cannot deal with that when you're using the Poisson or the logistic class of models to analyze some type of survival outcome, which is not time to event. So it's just something I thought you should know. And we said the constant hazard assumption is basically assuming that each covariate, each covariate, the effect of each covariate on the outcome or on the hazard function remains constant over time. Another way is to say that if you have, let's say, a variable like, let's say, gender, and we believe that the hazard ratio of male relative to females or vice versa remains constant irrespective of time, or remains constant regardless or independent of time, then that means that the constant hazard assumption was met with respect to gender as a variable. But situations where there are certain covariates that have time varying effect on the outcome. Example, if let's say you have a certain drug that might have a strong protective effect during the early stages, but loses efficacy over time, then it means a variable on efficacy might likely not meet the proportional hazard assumption. And then if situations where some variable interact with time, like for instance, age might have a greater effect on mortality, especially within, let's say, old individuals. So situations where, let's say, rate of mortality or the risk of mortality differs between older and younger population, for instance, having a variable on age is likely not to achieve the constant as an assumption. And situation where you are interested in disease progression, and then the disease progresses differently between different groups. So these are just a few examples to know in advance whether some covariates are likely not to achieve the constant hazard assumption. Now, I made mention of the parametric models where we said that for the non-parametric or the semi-parametric, it relaxes on parametric assumptions about the survival outcomes or the time to event outcomes. It doesn't assume it follows a certain non-parametric distribution. But situations where we believe that the survival time distribution follows a certain probability distribution, such as, let's say, Wellbo exponential or log normal, then in that case, we go in for the parametric assumption. So that is one of the instances. And there are also instances where we are interested in making survival prediction, or you want to predict what the survival time would have been at a certain future time. You cannot, although the classical survival models are able to estimate hazard ratios, they do not explicitly help you to what? Forecast or help you to extrapolate or help you to predict future survival times. So if you are interested in predictions of survival times, then the parametric methods becomes ideal. And situations where the constant hazard assumption is violated, then it means that one could also make use of the parametric survival models. But within the traditional survival models, you can consider the stratified analysis, where you stratify those covariates that violated that assumption. And that is also another alternative too. Instead of using the parametric, you can stick to the stratified versions. And situations where data is very sparse or sample size is too small, the Cox models, the estimates are much more stable with large sample size. Situations where sample sizes are also very small, which could occur, especially in rare cases like rare infections or rare diseases, where patient numbers or patient data are limited, then it means that parametric models might provide more reliable estimates. So this is something that, so these are just a few instances where parametric survival models might be ideal. So this is just summarizing it in a comparative sense. So it's just touching a bit on everything that I've said so far in a tabular summary. So once you get access to these slides, I think this will make life easier. So in summary, the standard survival models are flexible and mostly the assumptions are a bit relaxed. But for the Cox, we expect the hazard, the constant hazard assumption to be met. And for the parametric models, it can be used for predicting survival or for doing survival time predictions. At the same time, you impose some strong parametric assumption. But a disadvantage of that is if your parametric assumption or the probability distribution imposed on the survival outcome is incorrect or does not fit it well, then what that means is that it's not going to produce realistic or reliable estimates. Okay, so we don't have much time, but this was just going to be more like a case study where we are just going to investigate how to consider unstratified and stratified analysis and how to know whether some unstratified analysis needs to be stratified. So here we're just making use of a certain inbuilt R data called veteran data, and it's in a package called survival package in R. And this was just looking at, it was a clinical trial carried out on some veterans administration of on male veterans who advanced with lung cancer. And it was a randomized controlled trial where some individuals were either put into standard chemotherapy, which is like a control group, versus an experimental group. And the primary endpoint was looking at time until death. So it means that's a classical survival outcome. So these were just as examples of some of the covariates in that data. I won't go much into each of them. So these are the covariates in the data. So it means that suppose you want to fit a Cox-Proportional model to this. So the first thing is we do Kaplan-Meier analysis if you are doing any survival analysis. And when you do the Kaplan-Meier, Kaplan-Meier can only focus on categorical covariates. So you are comparing survival curves or survival times between categorical covariates. Okay, so it means if a variable is continuous, you may have to categorize it. Otherwise, you will have to consider it when you're doing the Cox regression modeling instead. And whenever you do the Kaplan-Meier curve visualization, you need to also investigate something called to statistically test whether there's significant difference between the survival curves. And a popular test we do is called the log rank test. And any insignificant p-value indicates that there's no significant difference in the survival curves or survival times between those the groups of the comparable variable under investigation. And one thing you should notice that anytime the curves intersect, then it will indicate that there will be no significant difference in the survival curves. So as you can see, anytime the p-value was insignificant for the log rank test, it was mostly because there was an intersection or the survival curves intersected at a point. All right, so that's just something I want you to note. And so now let's assume that we've now fitted a Cox-Proportional hazard regression model, considering all the predictors under investigation. And these are ways of visualizing the hazard ratios. And you can produce this same graph in R. So after fitting the model, the next thing is to ascertain whether some parametric, some assumptions, specifically whether the constant hazard assumption proportional assumption was met. And there's a statistical test to do that. And the famous test is called the Schoenfeld test. So it means that you can get the Schoenfeld individual test for each of the covariates. And anytime you see a significant p-value, that indicates that that particular variable, okay, violated the constant hazard proportional assumption. And so, for instance, it means that cell type in this case violated it because the p-value is significant, and so on and so forth. So it felt based on this Kano, which was looking at chemotherapy, the treatment group, and then I think the cell type violated the constant hazard assumption. So it means if I want to do such a study where I am still interested, so in this case, the Kano variable is like the treatment group. It's a variable of interest. But just because cell type did not achieve, or the Kano is a predictor of interest. But just as cell type did not satisfy the constant hazard assumption, instead of taking it out from your model, you can stratify your analysis with respect to that. How do you do that? So we'll look at that in the next 2 slides. So there are other assumptions you can actually also investigate. You can investigate whether there was outliers in your data, and after model fitting, and you can actually use the deviance residuals, and you expect the deviance residuals to be symmetric about 0 with a standard deviation of 1. That is to say, there should be some white noise in there. Okay, so, and there are some interpretations of that, and I won't go much into the mathematics here. Another thing is that the Cox-Proportional hazard model is a linear model, assuming that there's a linear effect of your predictors or covariate on the outcome. But what if some of the predictors do not satisfy this linearity assumption? So it means that there's also a way to visualize whether the assumption was met. So I've given some few interpretation of how to interpret or visualize instances where you are trying to detect for nonlinearity. And in that case, you visualize the martingale residuals and against your continuous covariate. Okay, so that's one thing you have to note. So the main remarks here is that whenever a particular variable, so let's say cell type did not satisfy the constant hazard proportion, proportion hazard assumption. So because of that, we have to stratify our analysis with respect to that variable. And to do that in R, the same function you use to fit the Cox-Proportional hazard model, you just have to put strata around a particular variable that did not meet that. But one take-home message is that when you stratify with respect to that particular variable that violated that assumption, just understand that you cannot make any inference on them. It is synonymous to when you are doing mixed effect modeling, where there are certain variables that introduces some variability to the data, but they are not predictor or fixed effect of interest. So you include them in the model as random effect. So, and when you include such variable that are, let's say, contributing to, let's say, temporal pseudo-replication in the data, that are contributing to, let's say, spatial pseudo-replication in the data because you collected the data at different locations and you believe that the location or region within the data was collected could have an effect on the outcome or could introduce some variability in the outcome, then you can include such a variable as random effect in the case of standard regression modeling. But when you do that, you notice that in those instances, we cannot make inference about the random effect variables, but we are trying to make sure we account for those variables that informs the kind of structure of the data we have, especially when the data is hierarchical or nested in some form. But those higher level or those higher nested level variables are not of interest to us, but we need to be able to capture them in the model. So with a stratified survivor analysis, basically those variables or the particular variable that did not satisfy the constant hazard proportion assumption, we stratify with respect to them, but we cannot make inference about them. And there are also instances where, because some predictors didn't have linear effect on the outcome, we don't still force to impose a linear predictive model, like still using the cost model to predict the outcome. So you can also use some machine learning survivor models based on random forest or classification tree to fit survivor outcomes. And in that case, you're going to end up getting something called a survivor tree. In R, there's a package called the Ranger or Ranger package that can help you to do that. So it means that this is a situation where some variables have non-linear effect on the outcome. So in view of that, you want to go for a more robust non-linear survivor model. And so this is how the survivor curves, if you fit those machine learning based survivor models, this is how you visualize it from, let's say, the random forest. So the blue is the average, and the average survivor curve will basically be that deterministic curve that you'll be considering as the survivor curve of interest. So it means it fits range of different survivor curves, and then you find the average. So this is how it is done. So let's look at a situation where I compared between a Cox model, Kaplan-Meier, a Cox model's survivor curve, a Kaplan-Meier survivor curve, and using a random forest-based survivor tree. And so this is just a comparison. So this is how they can, example of how they can be, they are comparable. So in summary, if some variables do not have a linear effect on the outcome, don't still impose linear model, or don't still impose or use the Cox model if there are better candidate models for that. Okay, so this is just a remark on the stratifications. Eventually, we only stratify the Cox model when some covariate did not achieve the constant hazard proportion assumption. And just that if you stratify, just know that you cannot make inference about them. And it happens a lot. So this is something we should note. And whenever some of the variables are also time varying or they change over time, then it means you should stratify with respect to time. So that is something you should note. Now, because of time, I'll briefly just introduce the multi-state Markov model. So you notice that for the standard survival models, let's say we are looking at time until death or time to death. So it means maybe some individuals were given treatment and want to see how they respond to the treatment. And in essence, we are actually looking at time till death or time till they die from that particular infection. Now, under such instances, you notice that we ignore the various infection history that an individual goes through before death occurs. So it means that over time, an individual was infected, could lose their infection, could get reinfected before they eventually die. But all those history are ignored. And we are just interested in, by the end of the study period, was the individual dead or alive? That's the event. And if the individual was dead, what was the time? Those who did not die, they were censored. That's basically what the classical survival models actually do. And that is a major limitation because in that case, it doesn't make those models very meaningful. Remember, a famous statistician called George Box once said, all mathematical models are wrong, but some are useful. So it's about how realistic and useful your model is. That should determine how relevant your model is to modeling the kind of data you have. But there are instances where those history could be, if those history can be neglected and it will not have any effect on the time to event outcome of interest, then in that case, the standard ones still suffices. But situations where, for instance, if you are looking at time to death and the disease progresses at different stages, and you are so meaning that, like, let's say HIV, an individual can move from stage 1 infections to, let's say, grade 1 or grade 2 infection. Or let's say diabetes, we have type 1 and type 2. And let's say an individual over time can switch from type 1 to type 2, from type 2 to type 1 before they eventually die. It might be interested, or it might be of relevance or clinical realism to capture that. And situations where we are also interested in investigating factors that predict transition from a particular state to the other, then it means that this multi-state Markov model extensively and robustly extend the classical model. So this is something I want you to note. And we have the time homogeneous case and the homogeneous, and the time inhomogeneous case. The time homogeneous case is when you are assuming that transitions between different disease states or different infection states or different states of interest remain constant over time. And if that holds, if that assumption holds, then that is a time homogeneous case that you are going to consider. But in real life or in real setting, hardly would the transition rate, okay, determine the rate of transition between different disease states or different states. Hardly would that assumption, hardly would that be time invariant. So most of the time they change over time. So we will look at a few examples of the multi-state Markov model. So it means in practical applications for those who are interested, just as I've said, anytime you are looking at disease progression studies, example, for instance, let's say cancer, we want to look at how cancer progresses over time. An individual was once healthy, they got to a localized cancer state, and then maybe from localized went to a metastatic state before death eventually occurs. Or maybe they can, at the metastatic over time, maybe can come back to localized. In that instances, using the multi-state Markov model will be better way to capture those different disease states before death eventually occurs. And there are relevant quantities, other relevant quantities that we're able to estimate. Even, for instance, what is the mean surgeon time, or what is the average time that an individual can remain healthy, can remain in a particular state? And you can even look at that over time. So it means it gives you more flexibility and more knowledge about the data that you want to investigate. And situations where you are looking at hospital readmission and recovery. So it means situations where an individual could be hospitalized, discharged, become hospitalized again, discharged, get readmitted, and get discharged, or eventually die over time. So this is to say that situations where you are interested in such kind of progression, whether about disease or whether about adherence or whether about anything. So in that case, it means that the multi-state Markov model becomes handy. So there's a particular inbuilt data called the CAV data, which was actually a certain form of cardiac infection from a heart transplant. Basically, it was a heart transplant which was tracking in the patients through different health states, ranging from healthy post-transplant conditions to advanced version of the disease or eventually death. So it was looking at about three or four different disease states, including death. And this is an inbuilt data that can help people who are interested in applying or adopting the multi-state Markov model to their respective studies to get a fair idea about how even their data should be structured to necessitate the kind of analysis. And there is this link of the articles that would help you to go through the various modeling and steps of the multi-state Markov model. In addition, I've already written a paper in a biological, a K1 biological journal, where I looked at deriving certain other relevant quantities in the case of the time-inhomogeneous case. So if you visit this particular link or you search my name and you look at a paper, you go to Google Scholar, you look at a paper on spatial and temporal parasite dynamics, you would also get to know most of the mathematics and how to derive explicitly certain relevant quantities that the classical survival models are not able to estimate. So if you look at this example, when we're talking about the time-inhomogeneous case, so this one is trying to observe, so there were three states. In that paper, there were three states, where state one was an individual, a host. In this case, the host was a fish, infected but alive, and there were instances they were alive without the infection, and there were instances they were dead. Okay, so or there was a dead state basically. So it means that here we are looking at transition from state 1 to state 2 and also from state 1 to state 3. As you can see, these observed transitions are changing over time, and these are based on the data. Okay, and and the package, the MSM package, which is used for multi-state modeling. So there's a package to do that in R, allows you to model or to capture the time-inhomogeneous case using a form of piecewise modeling technique. Okay, so this one is just demonstrating that the rate of transitioning from a particular state to the other was changing between different time points or over time. So if I should have used a time-homogeneous model, it means that that model was not going to have a very strong or very good predictive accuracy. So this is why you should always check whether time-homogeneous case or the time-inhomogeneous case is applicable to your data based on whether the transitions over time changes or whether the transition rates are time invariant or time varying. And these are other goodness of fit tests you can obtain from that. Yeah. So this is how accurate. This is just telling how accurate the multi-state was able to model this kind of data. Okay. So this, without wasting time, I will end here because it's supposed to be a short presentation. And so future BRC talk will look at multi-level modeling in clinical trials by a different presenter. And for those who are interested in learning R programming at the advanced level, or those who are interested in learning advanced statistical methods, I also have a YouTube channel called Clement Chumesi Educative Channel. If you just go to Google and you type Clement Chumesi, my name, you'll be able to get access to my videos. And this link also takes you to the channel directly. So I'll make these slides available through the organizers, and those who are interested can access it. So thank you very much. And at this point, I'll take questions. Thank you.